Hello, this is Dr. George Lisjak. This media sharing video presentation, uploaded to YouTube, is an integration of anatomy and physiology with microbiology, specifically the warfront between the lymphatic organ system and functional component, the immune system, versus pathogenic microbes, where bacteria are of primary interest in this presentation. We're going to examine both sides of the war between humans and these invading pathogens, including the principal cells involved, their strategies for conquest, both offensive and defensive. First, we're going to define a microorganism, or microbe. They are essentially microscopic organisms, especially bacterium, virus, or fungus. Types of microorganisms: I've broken down the types of microorganisms into two major categories: prokaryotes and eukaryotes. The prokaryotes are single-celled organisms that lack membrane-bound nuclei, mitochondria, or membrane-bound organelles. Eukaryotes, on the other hand, are any organism whose cells contain a nucleus and other structures enclosed by membranes. Of particular interest regarding the prokaryotes are bacteria and archaea. The bacteria are broken down into 6 phyla: Proteobacteria, Firmicutes, Actinobacteria, Cyanobacteria, Chlamydiae, and Spirochetes. Archaea are broken down into 2 major phyla: Euryarchaeota and Crenarchaeota. Eukaryotes we've got broken down into protozoans, fungi, and animalia. Protozoans include flagellates, amoeba, ciliates, and apicomplexans. Fungi are comprised of molds and filamentous fungi, and animalia consist of platyhelminthes, cestodes, and trematodes, as well as nematodes. Again, our focus is going to be on pathogenic microorganisms or microbes. In specific, with this presentation, bacteria. And bacteria constitute a large domain of the prokaryotic organisms. Okay, next we're going to move into the first line of the body's defense. So we're going to go from the invading pathogen side of the line to the human side of the line. The first line of defense is the external barriers, including the skin and mucous membranes. It is one of the 2 innate or non-specific defense systems, and are referred to as nonspecific resistance. Okay, and now we're going to switch back momentarily and define a few terms: pathogenicity and virulence. Pathogenicity is the ability of a microorganism to gain entry into a host and bring about disease. Virulence refers to the degree of pathogenicity. Virulence is derived from the Latin root virul, which means poisonous. Microorganisms gain entry into a human host by various ways. A portal of entry is the specific and characteristic route by which exogenous pathogens enter a host, and this varies between organisms. Various portals of entry include the respiratory portal, gastrointestinal portal, the fecal-oral route, sexually transmitted portal, and parenteral portals, such as piercing of the skin, mucous membranes, for example, cuts, bites, wounds, and injections. Let's define infectious dose. Infectious dose is the number of pathogens that need to be taken into the body in order to cause infection and possibly disease. Next, we'll go back to the second line of defense. Once the first line is breached, which again was the external or surface barriers, then the second line of defense kicks in. Again, the second line of defense is one of the two innate nonspecific defense systems referred to as nonspecific resistance. The second line of defense includes phagocytic cells, leukocytes or WBCs, white blood cells, and macrophages, antimicrobial proteins, immune surveillance, and inflammation. Inflammation is the hallmark of the second line of defense. And along with inflammation comes fever or increased body temperature. Okay, once we have breach of the invading pathogens into the human host, let's examine the insides of the human host, which deal with this invasion, and that is the lymphatic system. The lymphatic system is a network of tissues and organs that produce immune cells and vein-like vessels that penetrate nearly every tissue of the body, that recover tissue fluid, inspect it for pathogens, activate immune responses, and return it to the bloodstream. The components of the lymphatic system include lymph, lymphatic vessels, lymphatic cells, tissues, and organs. Lymph is interstitial fluid that enters into lymphatic vessels, is clear, colorless, similar to blood plasma but lower in protein. With its formed elements or cellular components, like blood, lymph is sometimes considered as a connective tissue type. The lymphatic vessels from smallest to largest include lymphatic capillaries, lymphatic collecting vessels, lymphatic trunks, and lymphatic ducts. We're going to spend more time with lymphatic cells, tissues, and organs. Lymphatic cells include lymphocytes, macrophages, and dendritic or accessory cells. Lymphatic tissues include diffuse lymphatic tissue, lymphatic nodules or follicles, and Peyer's patches located in the distal ileum. Lymphatic organs include lymph nodes, tonsils, thymus, spleen, appendix, and red bone marrow. Let's go a little bit further into the types of lymphatic cells. There are three primary types of lymphatic cells: lymphocytes, macrophages, and dendritic cells. Lymphocytes include natural killer cells, T lymphocytes, and B lymphocytes. Natural killer cells are large lymphocytes involved in immune surveillance that destroy bacteria, transplanted tissue cells, and infected or cancerous host cells. T lymphocytes mature in the thymus, later depend on thymic hormones. They are involved in direct attack and destroy pathogens. B lymphocytes, in contrast, become plasma cells when they leave the bloodstream and enter into connective tissue. They are involved in indirect attack, producing antibodies that immobilize antigens so that they can be destroyed. Macrophages, the second type of lymphatic cells, are phagocytic cells formed from monocytes, which are white blood cells that are agranular, that leave the bloodstream and enter into connective tissues. They destroy tissue debris, dead neutrophils, and bacteria. They display antigen fragments to T cells or T lymphocytes. And the third of the lymphatic cells types are dendritic cells, also known as accessory cells. These are branched mobile APCs, or antigen presenting cells. They're found in the epidermis of the skin, such as the Langerhans cells, mucous membranes, and lymphatic organs. They process antigen material and present it to T lymphocytes. They engulf pathogens using receptor-mediated endocytosis rather than phagocytosis. They act as messengers between the innate defense systems, first and second lines of defense, and the adaptive defense system, which is the third line of defense, which we will be getting into. Okay, let's switch back to our opponents, the pathogenic bacteria, and we are going to look into something called virulence factors. Remember, we defined virulence as the degree of pathogenicity or ability of microorganisms to gain entry into the host and cause disease. The virulence factors are structural or chemical factors that a microbe possesses that contribute to its virulence level. They are molecules expressed or secreted by pathogens that allow for their colonization in a host. Virulence factors include their ability to attach, immunoevasion and suppression, production of enzymes, both defensive and offensive, and the release of toxins, endotoxins and exotoxins. Virulence factors associated with attachment include adhesins. Adhesins are molecules attached to bacterial pili that allow for bacterial cells to attack host tissue. Another important concept or behavior of bacterial cells is biofilm formation. Although it's not considered directly a virulence factor, biofilm is a sticky layer of extracellular polysaccharides and proteins enclosing a colony of bacterial cells that resist body defenses and drugs. Let's look at immunoevasion and immunosuppression virulence factors. Immunoevasion, we have capsules and slime layers of bacteria. These are composed of a glycocalyx, a polysaccharide protein layer attached to the bacterial cell wall. And we have immunosuppression, specifically immunoglobulin protease, an enzyme that breaks down antibodies produced by the host. The third virulence factor is the production of enzymes. Again, there's defensive and offensive enzymes. Defensive enzymes include clot-forming enzymes such as coagulase, which forms a fibrin clot or coat over a microbe or colony. Offensive enzymes include clot-breaking enzymes such as the kinase enzyme used by strep and staph, staphylokinase and streptokinase. The kinase enzyme breaks down the fibrin clot or coat to facilitate pathogen invasion. And the fourth virulence factor concerns toxins. Some bacteria produce toxins. There are two basic types, endotoxins and exotoxins. An endotoxin is a toxic lipopolysaccharide component of the cell wall of gram-negative bacteria. These are released upon disintegration of the bacterial cell. Endotoxins are associated with increased body temperature or fever, body ache, weakness, and diarrhea. Endotoxins increase blood vessel permeability. therefore are associated with decreased blood volume and increased swelling. Exotoxins are heat-sensitive molecules produced during gram-positive and gram-negative bacterial metabolism. They are released into the environment or injected into a cell host. There are three basic types of exotoxins. Cytotoxins are exotoxins that kill cells. Two examples are leukocidins, leuko white, cidin kill, that makes holes in white blood cells, therefore destroying them, and hemolysin, hemo, blood, lyse, to create lysis or cell breaking, and hemolysins lyse red blood cells. Another exotoxin are neurotoxin. Neurotoxins are exotoxins that interfere with the host nerve cell transmission. The two most lethal neurotoxic exotoxins known to mankind are the tetanus toxin, formed by Clostridium tetani, and the botulism toxin, botulinum toxin, produced by Clostridium botulinum. And the third exotoxin group of exotoxins are enterotoxins. Enterotoxins are those exotoxins that affect the intestinal tract. Specifically, an example would be cholera, which produces the enterotoxin of which produces diarrhea. Then we're going to look into the invasiveness of pathogens or pathogenic bacteria before switching back to the human side of the battle between microorganisms, specifically bacteria, and the human organism. Invasiveness is the ability of a pathogen to penetrate and spread. One type of invasiveness characteristic of pathogenic bacteria is purposeful phagocytosis. Purposeful phagocytosis is where some bacteria allow themselves to be phagocytized by macrophages. Again, macrophages are monocytes or white blood cells that leave the bloodstream and enter into tissues and are phagocytic. By engaging in purposeful phagocytosis, where they are uptaken by macrophages, they can spread through the human body undetected by the immune system. Some bacteria, once they've been purposefully phagocytized, produce an actin tail. Once inside the cell host, they trigger the host cell to synthesize an actin tail, and this is used to propel them throughout the host cell cytoplasm. These cells can also jump from host cell to host cell, much like jumping from one train car to the next. And an example of a type of bacteria that engages in this kind of activity, causing it to be more invasive, is called the Listeria monocytogenes, which produces listeriosis. Okay, so let's switch back to the human side of the battlefront, and we're going to talk about the last line of defense, the third line of defense, which is the immune system. The third line of defense, or immune system, is an adaptive, specific defense system, including humoral referring to antibody immunity involving B lymphocytes or B cells, and cellular immunity involving T lymphocytes or T cells. This line of defense is characterized by specificity and memory. It is referred to as specific immunity. Okay, so there we have it as an outline of the battleground between the human organism and invasive pathogenic bacteria.